Well, hello everybody. I really hope you like microscopes, because you're going to be hearing a lot about them. So this is going to be a talk about microscopes and also about hair cells. And hair cells are part of your inner ear that helps you to hear and helps you to balance. I'm going to talk about both things. I'm going to talk about how the development of microscope technology has helped us to better understand how our hair cells work and how the ear works. So the first question is, what is a microscope? Now, I've got to say, when I made this slide, I had a bit of a panic because I thought, how many royalty-free pictures of microscopes am I going to be able to find? And then I thought, look in your own camera roll. So all of these are pictures I have wandered around taking of microscopes I look after. And a microscope these days is a lot of different things. So it was once a single tool. It was a tool that we use to make something that is invisible visible. And that's still what we're doing today, although the ways that we do that have expanded massively. We're only going to cover tonight a fraction of what is now used. And we're only really going to cover two main types of microscope. That's light microscopes and electron microscopes. And the thesis really of this whole talk tonight is about the way that discovery follows technology. In order for us to be able to make the scientific discoveries we make, we have to have the technology available. And this is going to be about how we follow from one to the other. So, I'm afraid we're going to need to do a little bit of microscope fundamentals. I am a biologist. I get extremely nervous when I talk about physics. So, we're going to have to talk about physics, but we're going to keep it as broad as we possibly can. And please be gentle. So, despite the differences, actually most light and electron microscopes are pretty fundamentally similar. Indeed, all microscopes are actually pretty fundamentally similar because they have a similar number of things that they need. They need a source of imaging particles. They need some way of concentrating those particles into a beam so we can use it for imaging. They need a sample to look at. They need an objective. And they need, which is going to do your magnifying, and then you need a projector, which is another set of lenses which is going to help you magnify. And right at the bottom of there, you can see there's a detector, and the detector is what we're going to see, what we're looking at. Sometimes that's the human eye, sometimes it's a photographic plate. These days, more often, it's some kind of digital detector. Some microscopes pass light through a sample. They're probably the ones that most people are familiar with. Other microscopes, they bounce light off a sample, and we'll talk about both types today. The other thing we need to have a brief conversation about is illumination, which is what these little diagrams on this side are about. There are two ways for the microscopes to talk about tonight that we illuminate samples. One is what's called whole field illumination, which is what's at the top here, and that is where we illuminate the whole area we want to look at, and we look at it all at once. The other way that we use it is that we can scan a beam across a sample. So we can have a much smaller beam and we scan it across. And that's going to become important later when we talk about how different microscopes work. So why do we use different particles? Why do we use light, or why do we do electrons? And this fundamentally comes down to the idea of resolution. Again, I am not a physicist. I get nervous when I talk about physics. So I'm going to explain this with some ping pong balls that I have stolen from my cat. Here is the cat. So the term resolution these days, as it's used commonly, quite often isn't used properly. And for this, I entirely blame mobile phone cameras. Because when we're talking about mobile phone cameras, we tend to talk about pixel number. This camera has 10 megapixels. But your pixel number isn't actually the main determinant of resolution. Your main determinant of resolution is your lenses and your imaging particles. So, this is an image of a cat. It was taken with a mobile phone camera. If we simulate what happens if we take the lens away, cat disappears. Without a lens, we can have the same number of pixels, but nothing is focused. And with no focus, all objects start to bleed into each other. So resolution is all about being able to tell things apart. And this is limited by focus, and it's limited by imaging particles, and this is where the ping pong balls are going to come into it. So I want you to imagine these two ping pong balls are two objects. If I hold them far apart, it's really easy to see that they are two things. But if I bring them much closer together, it becomes harder and harder. And if you imagine you were looking at this from the top, it would look like a single object. That's because the light can no longer pass through the gap between the two. Now, if I looked at this with an electron beam, electrons at high energies are smaller than visible light photons, and they would be able to fit through this gap. So this would appear as two objects. And that is essentially the basis of the idea of imaging particles and resolution. I should also say, I am going to talk a lot tonight about imaging particles. Okay, so I am going to talk about things that you can pretty much imagine as being a little ping pong ball. In reality, because reality is always more complicated, particularly in science, we should think of them both as particles and as waves. So when we talk about a smaller particle, we're also talking about something with a shorter wavelength. This is foreshadowing, because this is going to come up again later. Okay, so half of this talk is about microscopes. The other half is about hair cells, which means we need to talk about what is a hair cell. Well, a hair cell are these two enormous things over here and over here. These are cells that are mechanical sensors. They can sense movement, and they use that to sense hearing and for our sense of balance. Now, perceptually, when we're wandering around, those are quite different senses. They don't actually feel, for us, quite similar. But they're both actually about sensing motion. If we're sensing sound, we're sensing a sound wave. If we're sensing balance, what we're sensing is the movement of ourselves. Okay? So it's a wave, again, this time in the fluid. It's in the ear. You have 4 types of hair cells in mammals. You have your inner hair cells, which is this guy over here. You have your outer hair cells, which is the tube guy over here. Those are your 2 which are in your cochlea, and they are to do with your sense of hearing, and they're the main ones we're going to talk about tonight. You do also have 2 other types of hair cells, which are in the semicircular canals of the vestibular system, so those lovely loop shapes up there. Those are slightly less imaginatively named type 1 and type 2. Biologists are not good at naming things imaginatively. It's worth remembering that your inner ear is fluid-filled, okay? So if we take this diagram of the ear, you have your external ear, so the bit you can see. You have this air-filled region, which is the middle ear, and then you have the inner ear, which is this purple organ here with the little spiral. So that is full of fluid, okay? And inside that fluid is a spiral of cochlea, which if we cut it through the middle, would look a bit like this. And this is the part of the cochlea we're going to talk about. It's called the organ of Corti. Remember that name, it's going to come up again. The organ of Corti is where your hair cells are, and this is like a shelf of membrane that goes all the way around the cochlea, okay? And this is the part that vibrates when a sound wave comes in. Your organ of Corti contains inner hair cells. So this guy on the left here is a kind of flask-shaped cell. Somebody I won't mention who's here tonight refers to them as the pregnant lady cells. And on the other side, we have 3 outer hair cells, which are your tube-shaped hair cells. Your inner hair cells are the ones that are actually transmitting information to your brain. Okay, these are the ones that are taking those sound waves, transducing them into an electrical signal at the synapse, and sending them to your brain. The outer hair cells don't send sound to the brain, as far as we know, but what they do is they help to amplify sounds as we hear them. Okay, so they detect them and they help to amplify them locally to give us better ability to hear frequencies. And we are really good at this. Okay, so I'm going to give you a little demo. This is 2 tones for any musicians. This is 440 hertz, so concert A, and 445 hertz. And I just want you to tell me if you can hear the difference where they go. Okay? Anybody hear it? Hey, good. You've all got relatively good frequency discrimination. That 5 hertz is what's sometimes called the just noticeable difference. Okay, so when we talk about hair cells, we talk about these 4 types, but actually the 4 types all have a relatively similar body plan, although they have different functions. As you can see, there are 2 enormous hair cells at the front here. These are approximately 25,000 times the size they are in life, and you have about 3,500 of this hair cell in one ear, and you have about 11,000 of that. Now, to give you some idea, this is a model of the human cochlea. So those, 14,500 cells, plus all of the other cells, which are helping to support them and to keep them working, are all fitting in a space that's about that big, okay? This is why biology is fantastic and also really hard. Hair cells have 3 basic regions. The first one are the sensory hairs at the top. So these form the mechanosensor. These are the part of the hair cell that's actually going to sense an incoming wave and deliver that information to the cell. The second region, down the bottom here, is what I've called the synaptic or transmission region. This is where the hair cell is sending information out to the brain or receiving information back in. And then the third region, I've slightly unkindly called everything else. Everything else is a lot of stuff. Some hair cells can do some really interesting things. Some hair cells can move, for example. But also happening here is everything to do with being a cell. If you're a cell, you have got to be making proteins. You have got to be making stores of chemical energy. You've got to be creating and destroying, and that's all happening in this region. Now, hair cells are what's called terminally differentiated, which means they don't divide. The hair cells you've got are the hair cells you've got, and in mammals at least, you're not getting any more. Okay? The other thing about hair cells is they're really easily damaged. Hair cells are very highly adapted to what they do. They are really, really good at doing one thing, which is taking a signal and transmitting that signal. What they are not that good at is surviving everyday life. Being a cell, it's a bit difficult. If there are any scientists in the audience or people who live with scientists, that description might seem a bit familiar. So, we've talked about microscopes, we've talked about hair cells. We're now going to talk a bit about how we got from the first microscopes to the microscopes that we used when we first saw what hair cells looked like. So we're going to talk a bit about early microscope development. Hair cells in microscopes only really kind of appear in about the 19th century. But by that point, there's a lot of microscope development that's already happened. And they're already quite good. So we're going to go back in time a bit, and we're going to look at how they got to that point. We're going to talk about 2 things in particular. One is the compound microscope, and the other one is lens aberration. So, These two fellows up here, you might recognise the one on the left. This is Galileo. In 1609, Galileo took a brief break from astronomy and annoying the Pope and decided instead to make a two-lens microscope. And after he made this microscope, a man named Giuseppe Faber, for the first time, used the word microscope to describe it, which is great. He wasn't the inventor, though. He was actually the second person we know of. There's probably lots in between who didn't write it down. The actual inventors were about 19 years earlier, in about 1590. It was this man, Zacharias Janssen, and also his father, Hans. Okay? And they were the first people to have the idea to put these two lenses together to make what we now call a compound microscope. It's quite often credited to Zacharias as the invention. However, as far as we know, Zacharias was born in either 1580 or 1585, and the microscope was invented in 1590. So I will leave it up to you as to how much you think his dad might have helped. So what is a compound microscope? As I say, it's a microscope with two lenses, and it's the kind of microscope you've almost certainly either seen or used at school. Okay? There are two lenses on this microscope. There is the objective lens, which is that kind of turret-like thing, and then there are also lenses in the eyepieces. Okay? When you use this sort of microscope, you have two levels of magnification. Your objective magnifies your image, and then your eyepiece lenses magnify that magnified image. That means you can multiply or compound that magnification together, which is great. Brilliant. We can get things, we can see things clearly. Unfortunately, there's a bit of a problem, particularly in 1590, and that problem is what's called a lens aberration. So lens aberrations are all the ways that when we pass light through a lens, we distort it. There's 5 main types, and I promise we're not going to talk about all of them. They divide into 2 classes. The first is what's called chromatic aberrations, and this is caused by putting white light through a lens. The light is split into its component colours by the lens. Anyone who's ever held a prism out in a sunbeam and seen a rainbow, seen this effect. And those colours then focus in slightly different regions. And this tends to appear as coloured fringes around your image. The second type is called geometric. Geometric are all to do with spatial aberrations. We're only actually going to talk about one of these, because otherwise we'll be here for an hour and you will all be terribly bored. But this is what we're going to talk about, is called spherical aberration. So spherical aberration is what happens when your light hits a curved surface. And what we have at the top here is the ideal, the perfect lens. Your light hits it, goes through, all focuses at the same point. In a spherically aberrant lens, your light hits it, and the light that goes through the middle of the lens will focus at one point, and the light that goes through the edges of the lens will focus at a different point. So we're going to attempt our first demonstration here. We'll see how it goes. And we're going to do this using the world's most aberrant lens, which is a glass ball. Okay? So, please enjoy my glamorous assistant here, who, bless him, not only had to do this, but also had to build it. So hopefully, if this decides to work, and if we can dim the lights down, you should see a tree, just about, yeah, and a building. Now, these are some slides that I bought in a charity shop. Turns out 35-metre slides are quite hard to find, so these are somebody else's holiday photos. I don't know who, but it looks like they had a lovely time. When we focus with the glass ball, you can see that if we focus the tree, the building becomes fuzzy. If we go the other way and try and focus the building, the tree becomes fuzzy. That's spherical aberration, because different parts are passing through different parts of the lens, so we're seeing something different. Thank you. Now, I'm going to briefly digress. This is going to happen a lot. I do apologise. We can bring the lights up. Thank you. In order to correct, we had to invent lenses that were able to correct for chromaticity and for spherical aberration. And the story of the discovery of achromatic lenses is one of my favourite stories of research skullduggery. So I'm going to tell it because I think it's fun. Okay, so in 1730, a man named Chester Moore Hall works out how to make an achromatic lens. And he works out that if you take two lenses and stick them together, you can use one lens to correct for the other. You can correct for achromaticity. Brilliant. Chester, though, doesn't want anyone to know. So he has a brilliant idea. He goes, right, what I'm going to do is I'm going to ask two different opticians, one to make each lens, and then they won't know what they're doing. It'll be fine. And he does this. And those two opticians both go, I know how to do this. And they subcontract both lenses to the same man. A man named George Bass. Now, George, who is a very talented lens maker, has been asked to make two lenses that clearly fit together. He's not a stupid man. He goes, oh, it's an achromatic lens. And George, unlike Chester, he doesn't want to keep this a secret. In fact, he starts making them. And he tells another man called John Dolland. Now, anyone of a certain age may remember an opticians called Dolland and Aitchison a few years ago. They eventually bought out, I think, by Boots. But John Dolland was the founder of Dolland. And at the time he was told about the achromatic lens, he had just started a lens-making factory. So he goes off, he makes the lens, but he writes the paper. And not only does he write the paper, he writes the paper, he gets himself elected to the Royal Society, and then he patents the invention. So, scientists, do not put off writing the paper. Otherwise, someone's going to come along and they're going to make it, and they're going to make an enormous lens company. Okay. It takes about 100 years from that point for achromaticity to make it from lenses into microscopes. John Joseph Lister, who is the father of Joseph Lister of antiseptics, in 1830 creates an achromatic lens. And then, a few years later, a company a few of you may have heard of, started by a man called Carl Zeiss. He comes along and he makes, with Otto Schott and with Ernst Abbe, a fully corrected lens, corrected in all three colours for achromaticity and also corrected for spherical aberration. So by 1884, we have those lenses available for microscopes. But we're going to go back a bit first, because we're going to talk about the first people who saw hair cells. And the first person we're going to talk about is this man, Antonio Valsalva. So Antonio Valsalva was fascinated by ears, which I think we can all relate to. This was in the 1700s, so before a lot of these microscope inventions had happened, but he was the first person to describe the ear as having an external, middle, and inner compartment, and he was also the first person to describe the cochlea and the semicircular canals. And if you have a look at these images, this is from his 1717 book, they probably look quite familiar based on what you've already seen, if in the case of number 4, a little bit upside down. Other than that, he's done really well. Valsalva was also a really great, he was very much into practical experimentation. He left a note behind which said, "Gangrenous pus does not taste good." Now, what's amazing is that he lived to be 57 years old, and as far as we know, he didn't die of an infection. Yeah. We're going to move forward in time now. We're going to come to the 19th century, and we're going to talk about 3 anatomists. There were a lot of people doing cochlear anatomy at this point, but these 3 are, I think, quite key to understanding the story. The first one we have is Alfonso Corti. If you may remember this organ of Corti, so he managed to get the whole organ named after him. We then move to sort of about 1860s. This is Victor Henson. Victor only managed to get some cells named after him. There are some cells in the cochlea called Henson cells. And then by the 1880s, we're looking at Gustav Retzius. Now, Gustav Retzius, and I could be wrong, I don't think has a cell named after him in the inner ear, but he did manage some cells in the neocortex and, weirdly, the lines in the enamel of teeth. So he did quite well. we're going to look at what each of these anatomists produced when they were looking at hair cells. So this is, first is from Alfonso Corti, and this is from his 1851 work. And over here, we have a modern image of the organ of Corti, taken from a 2020 paper, just so you can compare what he saw versus what we know this now looks like. He got some fundamentals right. He's got that kind of spiral limbasis structure here. He knew that there were cells on either side. He's got something like an overlying membrane, and he's got something like a basement membrane. So he kind of got the gross anatomy quite well. Corti, about a year after publishing this, actually gave up anatomy because his father died and he became Baron Corti, which I think is the retirement plan we're all hoping for. By the time we get to Henson, he started to get these microscope improvements coming in, and he is starting to make something that looks more like the actual image. He has the inner hair cell, and he has the rows of outer hair cells. He's got these structural elements. So this two cells that seem to be forming a triangle, these are called pillar cells, and they're very important for holding the organ of Corti up. So if you imagine the organ of Corti is an organ that vibrates, and as it vibrates, it needs to be able to keep its own shape. So it has lots of different cells providing structural elements that are going to hold it together. He's also got better structure of what's happening at the base of the organ of Corti. So the organ of Corti sits on a basement membrane, it's sometimes called a basilar membrane, and that is the main vibrating part. So if you imagine it, you've got an organ of Corti here, you've got a basement membrane, you've got another membrane up above called the tectorial membrane, which Henson doesn't have, but there's kind of a suggestion that he knows it might be there. He's put in a dotted line like he's not quite sure, which, again, yeah, fair. When the basement membrane vibrates, it vibrates the organ of Corti, and the organ of Corti will vibrate in itself and also will vibrate against that top membrane. It's called a tectorial membrane. And that's what's going to allow those mechanosensitive parts, the stereocilia at the top, to move. Okay? And the final thing that Henson has sort of got is he's got this kind of tract coming up. You can see it here as this kind of looks like a series of dark, wiggly lines or circles. That's the nerves. So those are the nerves coming up to contact their hair cells. Okay? Ooh, yep. When we get to Retzius, he's pretty much got it as good as you can get. I mean, if you did this on a modern image, as you can see over here, it's pretty much as complete as a modern image can be. The only difference is we don't have artists doing beautiful renderings for us, so we have to make do with this sort of thing. He has now got stereocilia in as individual cilia. He's got lots and lots of different types of cells. He's named them. His nerve fibers are now nerve tracts. Remember, this was 1884. So at this point, the apochromatic lens, the lens that can correct for both chromatic and spherical aberration, is coming in. It also, he gave us a surface preparation image. So this is the image. If you imagine the organ of Corti we're seeing in cross-section is one unit, and this unit repeats again and again and again along the cochlea. So if you look from the top down, what you'll see is 3 rows of outer hair cells and 1 row of inner hair cells. And what Retzius shows us is exactly that. 3 rows, 1 row, and then this space in between, and that's actually the tops of the pillar cells. So Retzius at this point has taken us about as far as we're going to get with cochlear anatomy. And we're actually going to use this little diagram here from the rest of the talk to remind you where we are when we're talking about different hair cells. But at the same time, we have to talk about something else, and that's electrons. So I have to admit a little, tiny little bit of bias here. I'm an electron microscopist at heart. It was the first thing I used. It's what I used for all my early research work. They're great. So if I get a little overexcited, that's why. We've reached the end of the 19th century, and these new tools are starting to come along. Electron microscopes start to appear in the early 20th century. But we knew that, in theory, you could image with electrons before that. However, there was a major problem that we needed to solve. And that problem was, how do you build a lens for an electron? A glass lens won't work. If you put an electron into a glass lens, it will either pass through completely unbothered, or it will bounce around inside it forever. Electrons are difficult. They are small, they are charged, and they really like bouncing off things. They're really hard to focus. They also won't even pass through air. If you put an electron beam through the air, it will just scatter. So you need two things. Firstly, you need a vacuum. You're going to have to put this through a vacuum. The second thing you're going to need is some way of altering the electron's path as a lens would do, but without using physical materials. Now, in 1927, Hans Busch came up with the theory that it was possible to make an electron lens by using electromagnetic fields. So rather than using a physical thing, you're going to use magnets. Your magnets create your electromagnetic field, and your electrons, as they pass through that field, will be bent or they will be impeded. They will be able to be focused. Specifically, he found that you needed rotational symmetry, so you need to have your electromagnets in a ring. And he found that the mathematics that described that sort of lens were the same mathematics as described glass lenses, which was excellent news. And it was fairly soon after that paper, in 1931, that the first electron microscope appears. And this is a picture or drawing of it. It wasn't a very good microscope. They weren't really trying to make a very good microscope. They were just trying to prove that this theory was correct. It could magnify to about 400 times, and this was built by Ernst Ruska and Max Knoll. Relatively soon after this, however, Ernst Ruska had a bit of a rethink, and he decided to use the basis of the design of a compound microscope to make an electron microscope. And so in 1933, he produced his second design. Now, in this design, and my German is non-existent, to be clear, so this could say anything, but I do know how microscopes work. This design has a way of generating particles, a way of generating an electron beam. It has condensers, so ways of condensing that beam. It has an objective in order to magnify, and it has a projector lens system in order to magnify even more. This could magnify up to 12,000 times. Suddenly, we are in a whole new world of magnification. To give you an example, this is my TEM at the Ear Institute. This is circa about 2021. And you'll notice that it's a bit nicer, I'll admit. It's got a computer, it's got some nice things. But fundamentally, these designs haven't changed that much. Dealing with electrons is very much the there's only so many ways to skin a cat of science. You can only do things so many ways. Now, you may have heard of two different types of electron microscope, and it became clear really quickly after the invention of the first type that we've just discussed, which was a transmission electron microscope, that there was another design that would also work really well, and that was the scanning electron microscope, the SEM. It was invented in 1938 by Manfred von Ardenne, and I can promise you that most of the, or at least some of the most beautiful images you've ever seen in microscopy come from SEMs. An SEM works by a different principle. So if we take our TEM first, this is our beautiful TEM sample. In a TEM, we would pass a beam straight through it and down, and we would image underneath. In an SEM, we're going to scan a beam across this surface, and we're going to bounce electrons off it. And we're going to look at what electrons are coming back. I've got two example images here to show you what I mean. So in a TEM, this is an image actually of myelin. So in your nerves, you have this insulating wrap that wraps all the way around some of your nerves and helps with conduct better. That wrap is what those very fine lines are there. On the other side, this is the stinger from a bee. This was actually brought in because one of my colleagues pulled it out of her dog's paw in the morning and thought, I'll take it to work with me, handed it to me, and I went, I'll put it in the SEM. What else do you do with it? And this is what I mean about SEM images. They are beautiful. In general, SEMs have a lower total magnification than a TEM, but they can do something TEMs can't. They can image the surface of a thick sample. So because we're trying to pass a beam through our TEM sample, it has to be really thin. If it's too thick, those electrons are going to do what electrons do, start bouncing around inside it. And by really thin, I mean a few hundred nanometers. So for people who don't routinely think in metric prefixes, that's a few hundred billionths of a meter. Okay, it's about 500 times thinner than a human hair. For an SEM, we can put a thicker sample in because we are going to bounce things off the surface of it. And that makes looking at certain things a lot easier. And cell surfaces are fascinating to biologists because they tell us a lot about how a cell is interacting with its environment. For both kinds of electron microscope, though, we have to create a sample that can survive in a vacuum and being bombarded with high-energy electrons. And to do that, we have to prep that sample. We usually have to do what's called fixation, which is where we use chemicals. Formaldehyde is the one everyone's heard of. There are other ones we use as well, which will fix the sample in position and hold its structure. We then put heavy metal stains in to bounce the electrons. So the most common ones, I think still in use really, are osmium, uranium, and lead. Being an electron microscopist, it's fun. Then we have to get the water out. So if we put samples full of water into the electron microscope, they won't survive the vacuum. So we remove the water by replacing it with alcohols, usually ethanol, like some of you have been doing in the bar. And then we will then either replace those alcohols with a resin, which will then cure solid, so we can cut those incredibly thin sections, or we will dry the sample so there's nothing left. And we tend to do that using either a chemical with a very low surface tension so it doesn't damage the surface, or we use liquid carbon dioxide. So once we've done all that, we then have to prep it, we then have to get it in the microscope. And this is why there's this kind of maxim about electron microscopy, which is essentially that it's using very big things to look at very small, very dead things. In terms of EM with hair cells, they were a revelation in the study of the hair cell surface, of the hair bundles, and of the stereocilia. So we'll do a quick reminder about what these stereocilia, these hair bundles, are. They are these little bundles on the tops of the hair cells, and they're your mechanosensor. They're not hairs. They are nothing like the structure of a hair. And also, interestingly, they're not cilia either. They're actually a kind of modified microvillus. So the microvilli are the little things you see on the top of a lot of cells. You also have them in your stomach tracts. To give you some idea of why they are not cilium, this on the left is a cross-section of a true cilium. And you can see it contains these little sort of figure-of-8 structures. Those are what are called microtubules, and microtubules are a cytoskeletal element that make up cilia. Stereocilium, on the other hand, look very different. They're made up of these tiny little dots, and that is a different cytoskeletal protein called actin. And it's in actually a very specific array, which is one of the things we learned when we started using TEM. Now, coming in, we knew that the hair cells were the transducer. That had been worked out relatively early on by a lot of people. But what we didn't really know was how it was transducing that signal. We did know that it had two directions. So, if we take a hair cell bundle and we bend it this way, so the taller stereocilia to the shorter, that's what's called an excitatory direction. So that means that when we do that, we will create a signal. If we push it the other way, it's an inhibitory thing. So if we push it the other way, it won't produce a signal. And from that, there were three hypotheses about how these were transducing a signal into the cell. The first was the idea that there was a linkage between the stereocilia and the base of the cell. Now, when we started doing SEM of surface preps, we could see there wasn't a linkage there, so we were fairly certain it wasn't that. Secondly, there was a suggestion that there was some kind of asymmetry within the stereocilium itself, so somewhere in this actin array. Well, when we did TEM and we started looking at cross-sections, we couldn't see any obvious asymmetries. So the third hypothesis was that it was to do with linkages between the stereocilia within the bundle. This was a pretty seminal paper. It came out in the 1980s, and it showed two types of links between stereocilia. The first type, which is over here, are links which are between the stereocilia in the same row. So in all the stereocilia of one height, we have these links between them. These are called lateral links. And through SEM, we found out that these are in all types of hair cells. The second type of link, however, was one that was running between the shorter stereocilia and its taller neighbour in the next row. And it, in fact, runs from the tip of the shorter one to the shaft of the longer one. It was shown both in SEM and in TEM, and here comes that imaginative naming again. It's called a tip link. It was shown also that they ran in the excitatory direction. And this is where the conceptual leap is made. Those links could be gating a channel. It's a bit like if you imagine a hinged lid. So you've got a channel, you've got a lid over it. As you pull, you open the channel. And this synthesis has been shown to be true by a whole variety of methods. So we've destroyed the links, we've pulled on them with molecular tweezers, we've looked at what happens when you take away the genes that help to make them. And this is now accepted, really, as the method by which moving the stereocilial bundle is starting that signal transduction. We're going to slip back now to light microscopy, because SEM and TEM gives us brilliant resolution, but it only works in fixed samples. Like I say, very small, very dead. Light, though, is a lot more gentle. You don't need a vacuum for light. You can put light, or at least visible light, onto cells, and they won't immediately die. They will die eventually, quite often. They don't immediately die. So when we were thinking about how do we move from this purely structural thing to being able to look at cells when they're alive, the obvious answer is with light microscopy. But there's a problem. Those light micrographs that we've looked at earlier would have been produced with staining. So tissue, in general, unless it has a reason or has certain molecules in it, is colourless. It doesn't have anything. It's too small to be diffracting the light. So we needed a way to be able to see, but without staining, because for staining, you generally need to fix a sample. These days, we've got a few more options, but certainly then. So we're going to have to go back to physics again. I'm really sorry. And we're going to talk a little bit about refraction, okay? And more precisely, refraction index. So to give you a technical definition, refraction index is the ratio of the speed of light between air and a material or a vacuum and a material, okay? Taking that practically, it's about how much light is slowed down and bent when it passes through a material, okay? This, incidentally, is the thing that causes lens separations. So this has been causing all sorts of trouble for years. But biological materials, so things like cell membranes or cytoplasm or nuclei, have a different refractive index to the light around them that isn't scattered. And because of that, the light that goes through them is going to end up traveling at a slightly different speed, which, if we go back to thinking about light as a wave, is going to mean that those wavelengths are slightly out of phase. And that phase change is what we can use. So we can use prisms, annuluses, lots of clever things to make those phase change appear as brightness or as shadows. I've got two examples here of two different techniques. These are both actually the same thing. This is a mosquito spermatheca by Judy. Judy is here somewhere, so if you want to know what this is, find her. Phase contrast, this came along in sort of the 1950s. It was invented by Fritz Zwicky, who got the Nobel Prize for it in 1953. It should be pointed out, because she's quite often left out of the story, but the first production of a phase contrast microscope was actually by a woman called Caroline or Lily Bleecker. And it's International Women's Day, so I feel like we should definitely recognise what she did. Yeah, there we go. Later, another technique came along called differential interference contrast, or DIC. This uses a different sort of set of prisms to create your contrast, but it's the same basic principle underlying it. It's this idea of refraction. So at the same time this is happening, we're also learning how to detect better. So we have been using our eyes, or we have been using photographic plates. We're now moving into the age of being able to record things. And in particular, we can record things at high speed. Okay, and that's really important when you're dealing with cells, because cells are living in a whole different universe of speed to the rest of us. So we're going to talk now about what this live imaging showed us, and for that, we're going to have to talk a bit about outer hair cells. I said earlier that some hair cells can move, and that movement specifically, as far as we know, is the outer hair cells. Now, the reason why they do it is something we haven't really talked about yet, which is the idea of the cochlea as a frequency analyzer. So the cochlea has a structure that's called tonotopic. Tonotopic basically means that as you move along the cochlear spiral, different frequencies will stimulate the hair cells. So at the very base of the cochlea down here, we have very high frequencies or very high pitches. Right up at the apex, we have very low frequencies or very low pitches. Now, outer hair cells are definitely something to do with that system. And one of the things that sort of gives us that idea is that we know that we tend to lose outer hair cells first, before we lose inner hair cells. And outer hair cells are compared to the two, outer hair cells are the even more delicate one. When we start to lose our outer hair cells as we age in particular, we tend to lose them from the base first, which is why as you get older, you tend to notice you can't hear high frequencies as well. It's also those, if anyone's ever seen those things about like noises that scare off teenagers, that's what that is based on. The way that a cochlea is tonotypic is based effectively on where it's going to vibrate best. And this is controlled by a whole bunch of things. It's controlled by the stiffness of that basement membrane, it's controlled by stiffness in the organ of Corti, and it is also controlled by OHCs, outer hair cells. So by the 1970s, we knew that losing outer hair cells meant that you lost some of your ability to discriminate frequencies, and you tended to get hearing loss. But it wasn't quite clear how outer hair cells were involved until in about 1985, a really odd property of them was discovered. This was discovered by imaging live unstained hair cells with DIC and also injecting current into them using a microelectrode. Of the two, imaging is the easy part. The bit with the microelectrode is very difficult. I'm going to show you a quick video of what hair cells do in this. And for this video, this was actually taken by Jonathan Ashmore, who is also, I think, here somewhere. And it's possibly the most famous video. Cells can move. And only if they move, they can move in sync to a stimulus. They've got rhythm. Now, one cell doing that, I mean, it's pretty neat, but it's not terribly impressive. So what you have to imagine here is you have to imagine that there are 3 rows of these cells. And this is going to be happening over probably a small region of the cochlea. So suddenly, you've got a whole load of these cells all doing that. They're doing it together, and at the same time, they're getting feedback from their own stereocilia, potentially also from the brain, to tell them what to amplify and what not. And then it starts to become clear how this becomes an active amplification. Now, over time from this discovery, it was also found that there is actually a motor protein for this called prestin, and we understand now a lot more about what happens when this system is disrupted. But it all begins here with phase contrast and a really rather sweet dancing hair cell. Okay, we're now going to get into sort of the mid-20th century of microscopes. And we're going to start talking about how we moved from 2 dimensions to 3 dimensions. So every technique we've talked about so far is fundamentally 2-dimensional, okay? And by that, I mean that we take an image and we project it onto a screen, okay? Even things that look a bit 3-dimensional, like phase contrast or SEM, are still fundamentally projections. And there's a bit of a problem with projections, which hopefully we're going to demonstrate for you now, with any luck. Thank you, glamorous assistant. I really should have made you wear a tutu. Right. Are we ready? Okay. Oh, yeah. Now, hopefully what you can all see is a projection of the rather nice RI logo. So we can have a look now at what in the box is making that projection. I turn this lamp off, or we can put the lights back up, please. Thank you. Well, it's actually the UCL portico. The thing is, what we see in 2 dimensions is not necessarily what we're looking at in 3D. And this is really important, particularly for the organ of Corti, because the organ of Corti is a 3-dimensional structure, and it's a 3-dimensional structure that moves. So the ability to image in 3D became vital in order to understand what was going on. And this is where the confocal microscope comes into the story. Once again, we have to go back to physics. And we're going to talk, first of all, about an interesting little thing called the camera obscura effect. So, anyone who didn't build these as a child, I mean, I don't know what you were doing, but if you didn't build camera obscuras as a child, a camera obscura works on a really simple principle. And that is that when you reflect light from an object, light will always be reflected in all directions. But if you put a pinhole in the way, you will be able to get rid of all of that light apart from the light that's being reflected from a single plane. That plane will be in focus, so you get a plane of focus, and therefore you get an image formed on the back wall. In this case, obviously, the image is inverted because we are looking at reflected light. This was applied to microscopy by a guy with a wonderful name. His name is Marvin Minsky, which I think is great. And he invented the first microscope, the first confocal microscope, that used this principle in 1955. This version we're looking at here is actually his second one, which is the one he patented in 1957. Now, a confocal is a scanning microscope. So like an SEM, it's not going to image a whole sample. Instead, it's going to scam a beam across it. And the way that this works is that he passes that beam, first of all, through a pinhole to turn what in this was a zirconium light source, which is a bright white light source, into a tiny beam that he could scan. And then he shone that at a 45-degree dichromatic mirror. So a dichromatic mirror is a mirror that will bounce some wavelengths of light, but let other wavelengths of light pass through it. Now, this dichromatic mirror was bouncing these first wavelengths of light down, and it bounced it down into the through this lens and into the sample. Okay? Now, remember I said about light reflecting from a plane? In the case of a confocal, the light is reflecting from a plane within the sample. And when it's reflected, it will come back up through that objective again, and because it's reflected, it will be at longer wavelength. So now, rather than bouncing, it passes through the mirror, it passes through a second pinhole, which is going to get rid of any out-of-focus light, and you're going to get just that single plane at your detector. The pinhole is doing 2 things. First of all, it's giving you better signal, because all of that out of focus light is basically noise in your image signal. But the other thing it's doing is it's meaning that your light that you're seeing is coming from a plane of focus that is thinner than your sample. So what you can do is you can look at multiple planes of focus going all the way up through your sample. And when you stack those together, you can make a three-dimensional image. This is called optical sectioning, and it's pretty much the basis, I think, of almost all 3D light microscopy. Nowadays, we've improved a few things on this design. We tend to use lasers rather than white light sources for a few reasons. They're really bright. One of the problems with using a pinhole is you're cutting out a lot of your light. They're also collimated, so the light is quite easy to control, and they're monochromatic, so we can actually select which wavelength we're going to use. And that becomes really important when we talk about the next bit. So, confocal really comes into its own when another discovery is made. The initial discovery was made around about the same time, about 1962. And at this point, a man named Osamu Shinomura discovers a really bright fluorescent protein in a jellyfish. It's a really nice protein. It absorbs light in the UV blue spectrum and it emits it in green. It's named GFP, or green fluorescent protein. That discovery has about 30 years. It's very interesting. But in 1992, another scientist named Martin Chalfie discovers some really interesting things about GFP. He discovers GFP isn't like other fluorescent proteins. It doesn't need an enzyme to activate it. So a lot of fluorescent proteins, you need an enzyme system or protein system to cleave them to make them fluorescent. GFP only needs oxygen. If there's oxygen present, it's going to be fluorescent. This also means it's got a really interesting property, which is we can express it in almost any cell or organism. We need it to do two things. We need that protein to create the correct structure, so fold up correctly, and we need some oxygen present. If both of those, it will be fluorescent. And then a third scientist, and this is why science is always great when it's collaborative, called Roger Sien, discovers there's another really interesting thing, which is that you can take the structure of GFP and you can change it. And when you change it, you can make it so that it reacts to different light wavelengths and gives out different light wavelengths. So suddenly it's possible to make a palette of colours in the visible light spectrum. And if we're using lasers, we can select which wavelength we're putting in to get a different wavelength out. Those three share a Nobel Prize for chemistry in about 2008 for this. So, what can you do with a really bright fluorescent protein that you can express in most cells? You can tag other proteins with it, is what you can do. There's two methods of doing this. The first one is to tag with antibodies. So for anyone who's a little bit further away from school science, antibodies are those little proteins that attach really specifically to other proteins. So you can make an antibody and it will attach only to that protein. In the immune system, which is where they come from, they're used for tagging things for destruction, but we can make them against most proteins. So if you attack a GFP tag to your antibody, you can use it to find a protein in a cell. And that's how this first image over here mostly has been made. This green here that's lighting up that tectorial membrane, that membrane that's over the top of the hair cells, that is an antibody which is attaching to a protein within that membrane. There is a bit of a problem though, and that is, one, some proteins just don't want to have good antibodies. I don't know what the problem is, but they just decide they don't want to do it. And the other problem is that doing this sort of imaging, again, tends to require fixed samples. Certainly at the time. Nowadays, we do have some other methods to get around it, but you would have to fix your sample, so you can't do this on live cells. But there's another way around to do this, because we know that if we express this protein within a cell and we get it to fold, we can make it fluorescent. So what you're going to do this time is you're going to take your protein of interest, what you're trying to find, and you're going to attach your GFP tag to it in there. You're going to do it genetically. You're going to add the gene. When it's expressed with any luck, it will be expressed with that tag, and then you can follow it through a live readout. Now, I'm going to do an example of this, and we're going to move away from ears slightly to some pond life that we have at the Ear Institute. You might be wondering why we have pond life at the Ear Institute. Well, the good news is it isn't a flooding problem. We have pond life at the Ear Institute because these are called hydra, and they are tiny animals, and they've got tentacles like this, and their tentacles contain mechanosensitive cells. They use them for catching prey, they use them for sensing movement, they use them for moving about. But those cells have something similar to stereocilia on the top of them. And what we're really interested in is finding out about how those cells work, how similar they are to stereocilia. Now, these particular hydra are tagged with a protein that will be in some of those mechanosensitive cells. And because of that, we can image that protein live. So you'll see them move in bright field, and then if we change the illumination over to GFP, you can see these tiny dots on the tentacles, and those represent those cells. And that, again, is a brilliant way of being able to do things. It means that we can look at things in live cells, we can follow and track proteins as they move. Now, trying to explain what confocal did to biology in general, it's one of those things where the effect is so massive, it's almost too massive to see. So in order to kind of talk about it, I'm going to talk about one example, and we're going to dive briefly into the world of hair cell disorders. So confocal is now pretty much standard in any lab that's got even sort of a passing interest in imaging. But we're going to talk about a disorder that affects a bit of the hair cell we haven't talked about yet, the synaptic end. So if you remember, synapses, where all this signal transmission is happening. It's where we're sending signals, it's where we're receiving signals. We're going to talk specifically about the inner hair cell, so this is the one that senses sound, and for these cells, we're going to talk about sending a signal. So, a brief explanation of how these work. At the base of the cell, there are these nerve terminals. And the nerve terminals are attached to the cell by this thing called the synapse. And the synapse is where the hair cell is going to release a neurotransmitter, which will then be detected by the nerve terminal, which will then cause the nerve terminal to send a signal. To be able to release it, the inner hair cells have all of this neurotransmitter in these tiny little spheres, these spheres of membrane, of cell membrane. These are called vesicles, or neurotransmitter vesicles. And when a signal comes in, these come down, they fuse with the cell membrane, they release their cargo of neurotransmitter, which is what diffuses across the gap. If you think about it for a minute, with what stereocilia are doing, this is really impressive, because you don't stop hearing just because a sound has been going on for a really long time. Some of you right now might be wishing that you do, but you don't. Your hair cells are capable of what's called indefatigable signaling. They continue to signal. They also can start and stop really, really fast. You hear a sound almost as soon as it's made. And in order to do that, there's a lot of structure at these synapses, which is causing that to be possible. So when the stereocilia move, they open this channel, and this channel allows positive ions to flood into the cell. These are mainly potassium ions. What those positive ions do is they change the voltage of the entire cell. And by changing the voltage, they actually open other channels, these little channels down here, these red channels, at the base of the hair cell, and these are voltage gated, so they respond to changes in voltage, and they allow another ion, a calcium ion, to flood in. And the calcium ion is what signals I should be releasing neurotransmitter now. There's a big structure in these which you will not see at most synapses. And this big grey structure in the middle, this is called a synaptic ribbon. I realise it doesn't look like a ribbon. Kind of looks more like a beach ball when you see it. The name comes from the retina, which also has synaptic ribbons, and those ribbons actually do look a lot more like ribbons. What a synaptic ribbon does is it acts a bit like a storage tank. So vesicles are attached to this ribbon, and this ribbon is tethered at the synapse. So when that signal comes in, there are vesicles immediately there, ready to go. Okay, this isn't necessary at slower synapses. Then there's loads of other vesicles which are kind of hanging around in the cell cytoplasm, and they can refill the ribbon. So there is always a ready supply, and this is how this indefatigable signaling is happening. Involved in that is a protein called otoferlin. Now, otoferlin, like a lot of proteins, drew attention to itself because it was found in genetic studies of families with hearing loss. So geneticists go out, they look at families with different types of hearing loss, and they try to find within them the faulty gene, or more often than not, faulty genes, which are causing that hearing loss. In this particular case, they found some families with a very similar kind of hearing loss. They narrowed it down first to a chromosome and then to a gene, and that gene was called OTOF, O-T-O-F, and it codes for this protein, otoferlin. So now we know the protein, we could make an antibody against it. And if we can make an antibody, we can use confocal to find where that protein is within a hair cell. So in 2006, that's what this group of researchers did. They probed the hair cells with an antibody, and they found the otoferlin at the base of the hair cell in this synaptic region. They then used a similar technique, but this time with electron microscopy. So in electron microscopy, we don't use fluorescent proteins, we use very, very tiny particles of gold. Those will show up in an EM, and using that, we can look at these electron micrographs. These are the synapses. The synapse is this kind of fuzzy line at the bottom, and the ribbon is this dark round shape. And if you can see these little gold particles, you'll see that they're concentrated around the synapse and around the ribbon. That was pretty good evidence that otoferlin had something to do with synaptic release. And now that they knew that, they could then look at functional studies to try and work out what it was doing. What they found is that these hair cells have got loads of vesicles, but they're really bad at releasing neurotransmitter. And it was worked out that otoferlin is actually needed for vesicles to fuse with the synapse and to release their cargo. Now, confocal has been used for hundreds of similar studies like this. And the ability of mixing the genetics with the confocal in order to find these is one of the best ways of starting to work out and puzzle out how all these different genes are involved in hearing. So, we're going to talk about our last little bit of light microscopy. This is super resolution, and this brings light microscopy all the way up into the 21st century. Super resolution kind of does what it says on the tin. It's all about breaking that light barrier, that resolution that's maximum. If you remember all the way back, some point in the distant past when we were talking about ping pong balls, that idea of letting pass through. Now, you remember I said we had to think about these things as waves. The barrier which you meet is called the diffraction barrier, and it's to do with the size of the wave, okay? What super resolution techniques can do is they can break that barrier. So for light, it's a barrier of about 250 nanometers. And this table is just showing you how many different methods now exist to do this. So you've got 4 sort of different techniques and a load of different methods that they've been put into. And they rely on different things. So some of them are physical, so they actually physically make a light beam smaller. And they do that by using an interfering light beam as a kind of donut around it to make it smaller. Others are computational. So all they do is they do things like reassign all of those out of focus lights back to their in-focus origin. And there's been a multitude of papers in the 2010s, the 2020s, carrying on using these techniques. For example, they've looked at how calcium is moving by using live calcium imaging in live cells at the synapse. They've looked at specific proteins in stereocilia. They've even been looking at prestin and how it is in the OHC membrane. And with the ability to work in live cells and the ability to image in 3D, and the resolution of some of these techniques can get down to 50 nanometers, these are what are making a whole new set of discoveries about hair cells. Now, we're going to go back to electrons because I love electrons. So we're going to leave light microscopy now for the evening, and we're going to talk about electrons and what's been happening in EM in the late 20th and the first 21st century. So, like light microscopy, EM has moved into 3 dimensions. And for TEM, this has been done mainly through a technique called electron tomography. So tomography might seem like a familiar word to someone who's had a CT or they've had a CAT scan. And that's the same principle, only it's using X-rays. So if you had a CT, what you tend to do is you tend to lie in a tube, and there's a camera and an X-ray source, and it rotates around you taking pictures. Rotating it round gives you pictures at lots and lots of different angles, and then computationally, you can produce a 3D image. And CT was invented in the 1960s, but even then it was realized you could apply this to electrons as well. But moving an electron source and an electron detector is a lot harder. So the solution was to tilt the sample. So you take your sample and you tilt it along these angles. Samples, unlike patients, are a lot less likely to complain about you doing this. And as you tilt it, you can take images at different angles and reconstruct it computationally into 3D. And this is, I promise, the last digression. We're going to do a quick digression into computation, because just like everything else in the world, computers have made a massive difference to microscopy. And electron tomography is a really good example of this because it's so computationally based. And we knew it could work in the 1960s. So this is a 1960s computer. This is an Elliott 803. It was a really popular computer in the 1960s because it was considered affordable and small. And it was used in a lot of universities who really like things that are affordable and small. I happen to have an 803 expert at home, and I showed him how you reconstruct a tomogram and said, if you put this on an 803, how long would it take? And he looked at it and he went, days, maybe weeks. Now this is my not very powerful laptop. That can do the same computation in about 10 minutes. And really, this is a lot in technology development, microscope development. The knowledge that you can do something is quite often just waiting for other technology to catch up to you. And with computation, that was very much it. Now, this is where I get to talk about my own work. What do we use tomography for? So what I've been doing for the past, I don't want to think about how long, I've been focusing on using 3-dimensional EM to understand the 3D structures of hair cells and what happens to them when they're damaged. So in this experiment here, I was looking at noise exposure. Now these aren't the sorts of noise exposures that give you hearing loss immediately. Those aren't terribly common in nature unless you like frequenting explosive testing sites. But these are the sort that can cause damage over time, and those are everywhere, and we inject them into our ears with headphones. Now, this sort of hearing loss has been shown to involve, at least in part, those afferent terminals at the base of an inner hair cell. And it's been shown that this sort of noise exposure can cause those terminals to be lost, it can cause them to be damaged, and it can cause them not to work as efficiently as they should. But that kind of hearing loss is different to what you might think of, because it causes, we think, particularly perceptual difficulties. So it's thought to be involved, for example, in the ability to hear one sound in a noisy room. Okay, so this essentially is the science of why you can't hear people at restaurants. Now, using tomography, we have been looking at what happens inside those synapses after noise exposure. These are tomographic slices, so this is in our 3D reconstructed volume, we've taken a slice. At the top is our undamaged, and at the bottom is our damaged. And you can see they do actually look different. There's something going on there. But to really see it, you need to reconstruct it in 3 dimensions. When we reconstruct it, we find that these little purple dots, which represent those neurotransmitter vesicles, are much reduced in the noise damage. There's fewer of them, which means there's fewer things to be able to signal. However, what there's a lot more of is these blue and red objects, and these represent membranes which have been brought back into the cell when it starts recycling those vesicles. So what we think is that what's happening is that these cells are able to transmit and they're able to bring that membrane back in, but they're not able to recycle it back into those vesicles. and that's why we're seeing this difference. Now, I'm going to talk about what happens because electron tomography has one big problem. It's great, but it only works on thin samples because it uses transmission EM. And in fact, the problem becomes even worse when you tilt a sample, because if you imagine a beam passing through here, as you tilt, you're going to be passing through a thicker and thicker region. So it's limited down to those few hundred nanometers. You can take multiple slices and do tomography in all of them and put them together, but the aberration in that is a nightmare. So the field is really looking for a way of doing the same thing, but with thick specimens. And when you think about thick specimens in EM, you think about SEM. Now, this is an image from an SEM. If you've been looking at the TEM images we've seen this evening, you might think that looks like a TEM image. And actually, we have done this at conferences with full of electron microscopists of playing Guess the Instrument. We're amazing at parties. The way that this is created in an SEM is you take a resin block of tissue, like you would use to section for TEM, and you scan the surface of it. And when you look at that, you get an image that looks a bit like this. But because you're imaging the surface, it doesn't matter if it's thick. And if you can then find a way to remove that surface while it's still in the microscope, you can image the next layer down, and the next layer down, and the next layer down, and you can make a three-dimensional image. There's a couple of ways of doing this. You can physically put a knife in the microscope. You can ablate that surface with ions. You can do your cutting outside the microscope and then put your sections in, which means you can look at things again and again and again. But they all come down to this very fundamental idea. So I started with these techniques because I wanted to understand about how hair cells are structured. And particularly, I wanted to understand inner hair cells. You may have realized by now, inner hair cells are my favorites. I wanted to know how you structure a cell to be that highly adapted to its function. And I was really interested in how they support their synaptic function, that indefatigable signaling. So I was looking at two different cellular organelles. So these are the organelles that are within cells. I was looking at mitochondria. Most people know because they produce ATP. And also the membranes, which are internal to a cell. And I was looking at them in the compartment below the nucleus. I could see, when I looked at them on these 2D planes, that there's some kind of asymmetry. So they are these little wiggly lines pointed to with that white arrow. But to really appreciate it, we had to reconstruct an entire cell in 3 dimensions. And when you see it here with its nerve terminals, you really start to appreciate that there is a big asymmetry of these cells, with all their membranes and mitochondria towards this side, which is also where most of your nerve terminals are. The other thing that we found when we did this, so mitochondria and internal membranes are quite often associated, but in hair cells, about 90% of those mitochondria are sitting on a membrane. And that's unusual. We knew that one thing that hair cells can use their mitochondria and their membranes for is for moving calcium between the mitochondria and the membrane. So they can use it to control their calcium stores. Now, you might remember that calcium is really important in synaptic vesicle release. And we knew that they did that by using little linkages between the mitochondria and the membrane. Those linkages are way too small to see by VEM. But you can see them by electron tomography. So again, because we knew where to look, we could combine those techniques and show that those linkages existed in here. And this study of combining techniques, imaging across scales, combining the best part of one technique with another, is really kind of the basis of where microscopy is probably going. So I'm going to talk about one more thing. This is the last imaging story. And we're going to look now at the imaging of things that are even smaller, the membranes or mitochondria. We're going to talk about imaging proteins. This is protein structure imaging. This technique is quite often called cryo-EM. That is not technically correct. Drives me mad. Cryo-EM is a technique for taking a tissue or a cell and freezing it so we can look at it in the electron microscope. And the way that it freezes means that all the water that is around it remains in the same position. It's frozen that quickly. And it freezes it in a completely lifelike state. Now, because it's frozen, we can't put any stains in it. But you can do phase imaging in an electron microscope just like you can in a light microscope. And phase contrast actually works really well with frozen samples. So this technique is actually called single particle cryo-EM. And it combines that ability to get things in a really lifelike state with being able to image at the level of protein structure. Previously to this, protein structures were mostly found by a technique called X-ray crystallography. And to do that, you need to make a protein crystal. It works brilliantly sometimes. A lot of very important work's been done that way. But sometimes your protein just won't crystallize. And sometimes your protein will change shape when it's crystallized, particularly if you're removing all that water. So for cryo-EM, it's very simple. You simply take a concentrated solution of your protein, so you've got as much protein in there as you can, and you freeze it. And once you've frozen it, you have a look at it in a transmission electron microscope. Now, when you've frozen it, all those particles, because they were moving about randomly, will have been frozen in random positions. Okay? So what you're going to get is lots and lots of pictures of the same thing at different angles. And it's pretty conceptually simple to see that if you give that to a computer, it should be able to work out what the 3D structure is that gives you all of those different angles. And that is essentially how this works. This won a Nobel Prize in 2017, and it's pretty much the staple of structural biology now. But how is it used in hair cells? Well, when we came into the 21st century, there's still a big question left in hair cell biology. I've talked about these tip links that gate this channel on top of the hair cell, but we didn't know what that channel was. There are a lot of candidates that have been suggested. It had been established that it's, rather than a single protein, more likely kind of a complex of proteins. And one of those proteins was probably acting as the pore, so the hole through which these ions are passing through. In 2018, there was a paper about a protein called TMC1 and its relative TMC2, and they used fluorescence confocal to show that that protein is at the tip of the stereocilia. They used live imaging with a fluorescent dye, and this fluorescent dye passes into hair cells via that mechanosensitive channel, and they showed that if you deleted TMC1, it didn't get into the hair cells very well. And they also used structural prediction, so they tried to predict how this protein folded up to see if it would become a pore. Now, structural prediction is really hard. Like, Google have been working on it hard. And it's really difficult to judge how accurate a structure prediction is because there's lots and lots of factors that go into how a protein folds. So enter single particle, and also enter these little guys. So these are C. elegans, the biologist's favourite nematode worm. Now, getting purified TMC1 protein complexes from mammals is really difficult. But it turns out that these guys also have TMC1, and it's acting as a pore. So in 2022, TMC1 and two of its accessory proteins called CARM1 and TMIE were purified from this worm, and they were examined by single particle. And the result they got showed a pore. That the TMC1 in the centre is forming that pore in the middle, and that these other two proteins are supporting it. This doesn't prove that this is what's happening in mammals, but it's some pretty good evidence. We know mammalian TMC1 is very similar to the worm protein. We can use the structure of the worm protein to try and better fold our predicted models. So now there's work continuing to try and get to that mammalian TMC1 so we can finally work out for certain what this is. So I'm going to summarise this talk by bringing back the phrase I used at the beginning. Discovery follows technology. This timeline shows at the top in green all of those microscopy discoveries we've talked about, and at the bottom in blue, all of the hair cell discoveries. And it's pretty clear that every time there's a flurry of microscopy technology, we start discovering new things about the hair cell. And I think that's where I'd like to end this, which is on the idea that we're going to find out even more stuff tomorrow because we're developing more technology, we're using more technology, and it's getting us closer and closer to understanding things. Okay, thank you very much.